Good afternoon, everyone. We're excited to have you all join us today for our final seminar series. We have Dr. Kang, who will be speaking. Before she begins, I just wanted to share a couple of announcements with you all regarding our seminar series for today. So I'll just share my screen briefly with all of you. So again, as a reminder, we'll have a majority of our session will be Dr. Kang's talk, and at the end, we'll leave time for Q&A. And to submit your questions, you can use the Q&A feature here at the bottom of your screen. And Dr. Kang's talk is Hpv from tumor virus to head and neck cancer epidemic. So just to briefly introduce Dr. Kang, she is a radiation oncologist who specializes in using advanced techniques to provide highly effective, precise, minimally invasive, and personalized cancer treatment. And she completed her undergraduate studies at Yale College with a degree in molecular biophysics and biochemistry, and went on for her graduate and medical school training with an MD PhD program at USC in Los Angeles, where she received her PhD in systems, biology, and diseases, and research in renal physiology. Her residency training was in radiation oncology, and she researched prostate cancer. Currently at MSK, she researches head and neck cancers with current interest in big data and oropharyngeal cancer and personalized medicine. Again, we'd like to welcome Dr. Kang for today's talk.
Perfect. All right. So hi, everyone. Thanks so much for having me here today. I'm going to be talking about the human papilloma virus. And I know this is the last in your entire series of lectures, but I will start with the basics just to make sure we're all starting on the same page. So question number one, what is cancer? And cancer is a constellation of many, many different diseases. Most of you probably know someone who's had cancer, and we typically name it by whatever organ that the cancer started in. But even though you could say there are hundreds of cancers, really a cancer cell is basically a normal cell that has acquired abnormal growth properties. So normal cells grow. They divide and grow. It's a normal thing, but there are a lot of rules, regulations, and signals that they have to obey in order to go through that growth and division process. But cancer cells don't listen to those rules. They do not follow the regular rules, and that's what makes them abnormal. They also have a couple special qualities. They can hide from or escape from the immune system. The immune system is supposed to be able to recognize when a cell is abnormal or not doing the right thing, but cancer cells have acquired a special ability to oftentimes escape immune surveillance. And another thing cancer cells do is they look different. They don't stay in line. They don't follow the normal boundaries of where they're supposed to be located or not. So on the left, what you can see is various ways all of these things manifest, with the left-hand column being normal cells. You see how neat and organized they are. They're very uniform. But you see on the right column, basically none of that is true. These things are big. The nucleus is big because there's a lot of DNA changes and replication going on. They are not uniform. The cells kind of look different from each other. They're disorganized, and they don't have very clear boundaries. All of this ultimately happens because there are genetic alterations. So cancer is a disease of genetic alterations. And sometimes we know which gene went wrong, and sometimes we don't. One cause of cancers that is probably not recognized as much as it should be is that viruses can actually cause cancers. And about 15% of cancers are associated with viral infections. So not the majority, but certainly a lot. And the thing that makes cancer the scariest the thing that we all worry about that we don't want, is they have a very special property that normal cells don't have. They can move. Normal cells, for the most part, stay put. They are grounded and cemented to wherever they were born. But cancer cells, blood cells, and immune cells are basically the only cells that have the capacity to move, to leave where they started. And that's really what makes cancer dangerous, when it can travel. So what is a virus? A virus is an even simpler thing to explain than cancer. A virus is a small connection of genetic material. That genetic material is either DNA or RNA, and it has to be surrounded by a coating that protects the genetic material. And that coating is typically protein. There could sometimes be some lipid or fat in there, but it's always at least protein. So a virus is just an infectious agent, a tiny microscopic infectious agent that's composed of genetic info and a protein coat. And you could see that on the picture on the right-hand side at the very top. So viruses, that's all they've got. So they don't have enough stuff. They don't have enough machinery to be able to make more of themselves or to make anything. They can't make proteins, they can't do anything. They basically need to parasitize or take over a living cell in order to have the cell do its dirty work for it. So viruses themselves, if there were no cells to provide the housing and machinery for viruses themselves really couldn't do much of anything. They really need to take over the cells. And there are nonillions of viruses, but almost none of them are known to actually even be able to cause disease in humans. It's estimated that only about 200 or so viruses of the nonillions are even able to infect humans. Even fewer are able to actually cause cancer. So there are 8 viruses that have been associated with human cancers. There are other viruses that have been associated with cancers in animals, but when we're talking about human beings, there are only 8 viruses that have been associated with human cancers. I'm not going to talk about all of them. I'm really only going to talk about HPV, human papillomavirus, the one up top, because it's the most common one that causes cancer, because it causes multiple different kinds of cancer. But this is just a chart for you guys to see if you're ever curious or interested in what those other viruses are. So, bottom line, if viruses can cause cancer, is cancer an infectious disease? So when we think of an infectious disease, we basically think of something that's contagious. And cancer is actually not contagious. So what we're going to learn really is, how is it that viruses can cause cancer? And what we're going to see is it's actually pretty inefficient. Even if you get infected with a tumor virus, a virus that we know causes cancer, it's actually a pretty rare event that will actually lead to a cancer. But it's still an important cause of cancer to understand, because viral infections are in many cases preventable. So if someone told you that, you could prevent a cancer, most people would say, I will do what it takes to prevent a cancer. We have 3 main objectives for the talk today. So the first part of the talk, I'm going to give a basic science background on tumor viruses. A lot of this research and work was done basically in animals. So we learned it from lab experiments, tissue cultures, cell cultures, and animals before we really fully understood it in humans. But it's important to understand these principles of how tumor viruses can cause cancer, because it can help us understand when we think we should be worried about viruses causing cancer, and it can help us understand how we might be able to fix or treat cancers caused by viruses. So this is a really nice long timeline. It's about 110 years covering some of the history landmarks in tumor virus research. I highlighted a couple of the interesting points, which is the first tumor virus was actually discovered when they first found that a tumor virus could cause cancers in other animals if you kept injecting them in 1908 by Peyton Rouse. And he's actually a scientist who did his work right here locally in New York City at the Rockefeller Institute, 1909. But he didn't get a Nobel Prize until 1966. A long time later. So oftentimes when people discover very important, interesting things, it takes decades for the scientific community to really recognize it and appreciate it. The first human tumor virus that was discovered was the Epstein-Barr virus, EBV. That was in 1964. It was implicated in Burkitt's lymphoma, and EBV is the same virus that causes infectious mononucleosis, or mono. And then another important sort of landmark, in my opinion, is 2006. That's when the FDA, the United States FDA, approved the very first tumor virus vaccine. It was the HPV vaccine. And that didn't happen until 2006. So you can see how long it takes for progress to be made. So going back to Dr. Rouse, Peyton Rouse, a little bit, what he discovered was a chicken sarcoma virus. So a sarcoma is a type of tumor that started in muscles. And so in this particular case, there were tumors from the breast muscle of a hen that he could basically cut out, grind up into a very, very small homogenate, and filter it through like a sieve or a filter that basically should only let the teeniest, tiniest particles through. So definitely bacteria would not have been able to get through. And what they found was that liquid that should have had almost nothing in it, if they injected that into an animal, that animal could get the same tumor in the breast muscle. So the ground up filtered liquid could cause a tumor. Now, to really prove that, what they did next then was actually they took the tumor from the animal that had the filtrate, that liquid injected, and they took out that animal's tumor, and they did the same thing, ground it up, filtered it, and they saw, well, can the induced tumor induce another tumor? And the answer was yes. So it does seem transmissible. And you know, the amount of virus that they could quantify in the filtrate was a lot more than what they injected. So they inject a little bit, but they see a whole lot once it's in the tumor. And that means there's replication, growth, and amplification essentially happening. So because I know that many of you are probably doing some lab work in school or interested in one day doing some lab work to do scientific experiments, I tried to put in a couple pictures of what we do in the lab so that you can get a feel for how we learned the things that we learned about these discoveries. So, I told you in the beginning that cancer cells behave abnormally. They basically don't listen to the normal growth rules. They grow however they want. They can travel, they can move, and you can actually see that under a microscope. So if you look at the three pictures, the blue pictures on top of each other in the middle of the screen, normal cells are very neatly organized, and they make something called a monolayer, which is they're going to be near each other, but they're not going to pile up on top of each other. They want their space. But when you infect cells with the RSV, the Rouse sarcoma virus, and if that cell becomes transformed, starts behaving badly, then it's going to grow uncontrollably. It grows right on top of each other and makes a very big pile. And you can see that on the microscope. So if you look at the gray panel on the right, what you'll see is, I'll give you a hint, two pictures show normal cell monolayers, where cells are near each other but not piling on top of each other, and they're very neatly organized, obeying all the growth rules. two pictures show, oh, those rules are broken. The cells are piling on top of each other and going all over the place. I don't know if I can hear the students, but I'm going to give you a moment. Normally in my talk, I would ask people just to shout out the answer. But which panels do you think show normal growth, meaning contact inhibition? Once they're near each other, they stay away, they keep their distance and create a monolayer. And which panels show abnormal growth or transformation, and now they're piling all on top of each other? So I'll let you look at these pictures, and I will give you a clue that the two pictures on the left, the ones on top of each other, are showing the same thing, and the two pictures on the right are showing the same thing. The cells on the left, the upper and lower on the left, are normal. Those are making a monolayer. They have contact inhibition. They have not piled on top of each other. But what you see on the right is the cells are a mess, all on top of each other, different sizes, very disorganized. So you know that that's where transformation, those cells are, they look like tumors. You know, in order to cause cancer, they learned that the virus, when injected into the cell, will cause the cells to grow abnormally. But it's not a hit and run phenomenon. It's not like you inject a virus, it definitely becomes a cancerous cell, and then it's like that forever. You actually need a lot of the times those viral infections won't cause any of this, but you need to have a lot of virus around, and it needs to have more opportunity and more time to eventually cause transformation. So one of the really elegant ways that this phenomenon was discovered was this. Normal body temperature is 98.6 Fahrenheit, or about 37 degrees Celsius. So normal cells are happy at that temperature. And this, they created a RSV viral mutant that also wants to be at normal body temperature, right around 37 degrees Celsius. Okay, if you get a very bad fever, let's say about 106 degrees Fahrenheit or about 41 degrees Celsius, normal cells can handle it. They don't like it. I mean, nobody wants a 106 fever for a very long time, but they can handle that amount of fever. This mutant virus doesn't like it. If it's 106, it basically is going to go into hibernation. It's not going to replicate. It's not going to divide. It's not going to do anything. It's just going to go into hibernation and wait for the temperature to come better. So what they found is if you take normal cells and you infect it with this RSV mutant, as long as the temperature is 37 degrees, you see that the cells look abnormal. They have the abnormal growth patterns. If you heat up the temperature to 41 degrees Celsius or 106 degrees Fahrenheit, all the cells came back to looking normal because the virus basically went into hibernation and it didn't want to do much on the cells. If you then cool the temperature back down to 37 Celsius or 98.6 Fahrenheit, then now that the virus is happy with that normal body temperature, it transformed the cells again. So this was sort of the way that they understood that it's not like you have the viral infection once and the problem is there forever. The virus has to hang around for a good long time for it to be able to transform the cells. So Ralph sarcoma virus was one of the first ones discovered, but there are a whole bunch of other viruses that have been discovered and are able to cause cancer in animals, both DNA-carrying viruses and RNA-carrying viruses. I won't go into detail, but this is just for you to see that many different viruses have been found to cause cancer in animals. And I told you that the temperature experiments showed that the virus has to stay around. It's not going to come in once and leave and still be able to cause cancer. It has to stick around a long time. Well, how is it sticking around? it basically has to find a way to integrate into the cell. And if you look at these pictures of the cells, what you can see is the round thing in the middle of the cell is the nucleus, and the round thing in the middle is what's lighting up. So that's where the viruses are integrating. They're integrating into the DNA of the host cell. So I'm not going to go into too much detail about this. This is really more for lab experimentation, but the basic important things to understand is that viruses have to integrate into host cell DNA in order for their the viral DNA to be copied, for them to make their proteins, to do what they need to make more of themselves. And it's actually very inefficient. So how successful is it that viruses are able to integrate into the host cell DNA? Not very successful at all. This is successful in less than 1 in 1,000 times. So the vast majority of times, viruses don't integrate into the host cell DNA very well. But when they do, then those cells can replicate a lot. And when those cells with the viral DNA integrated into them replicate, they're not making 1 or 2 copies, they're making hundreds. So when a virus, integrates into host cell DNA, there are a couple different ways that it can cause cancer. One is that the virus itself carried a tumor-causing gene. So the virus has, DNA or RNA code that actually codes for a tumor protein. That's what RSV had. It carried a tumor gene. It carried something called the Sarcoma oncogene. Another way that viruses can induce transformation and cancer is when they carry, viruses can take, as they integrate into the host cell DNA, in and out and in and out, sometimes they can actually take oncogenes that the cell already had. So it's been studied and found that not only do viruses carry their own unique oncogenes, cancer-causing genes, but viruses can actually acquire oncogenes from cells. Like normal cells, our normal cells can have genes that can cause cancer, and the virus can take that and produce that too. And then other times the virus can not carry an oncogene, but it can integrate into the DNA and cause something fishy to happen. So there are all sorts of different ways that viruses induce genes and genetic changes that can cause cancer. This is a very long list of all the proto-oncogenes or cancer-causing genes that have already been found. So these are genes that are found in normal animal DNA. So there are 30 vertebrate proto-oncogenes that have been discovered. So these are cancer-causing genes that are already inherently a part of these vertebrates. And so, if normal cells already have genes that can cause cancer, and if a virus can activate those oncogenes, they can steal those oncogenes, or if they carry their own oncogenes, those are all different ways that you can actually trigger transformation and cancer. If cells already have oncogenes, normal cell DNA has an oncogene, didn't need a virus. That's normal cell DNA was born with an oncogene. Do you think it is possible that that there are things other than viruses that could activate or turn on those oncogenes? It doesn't have to be a virus, does it? And there are other things other than viruses that can turn on proto-oncogenes. Viruses are not the only thing that can cause cancer in the genes that we already have that are cancer-causing. So I won't go into detail about this, but this is just an example of a virus integrating into the host cell DNA. Most of the time, it's going to integrate somewhere randomly where it's not going to cause a problem, but then occasionally it's going to integrate somewhere at a proto-oncogene, and that's going to cause cancer. And these are other cellular proto-oncogenes that have been found as well. So we said that when tumor viruses cause changes in the cell, there are a whole bunch you could see, like loss of contact inhibition. They're growing on top of each other. They're very irregular in shape, and all sorts of things happen. At the bottom line, it can look like a cancer, but you have to know, does it actually behave like a cancer? And so ultimately the test for tumorigenicity, if the virus actually really did cause a cancer, is to take those transformed cells and you have to inject them into a host and see if the host gets cancer. Okay, so hopefully, you guys agree that viruses can absolutely cause cancer. Viruses have actually also been used and are still used in making vaccines. So then the question is, can viral vaccines cause cancer? Because this is something that comes up a lot these days. Well, there's a reason that there's this controversy. Back in the 1950s, they were generating poliovirus vaccines. And they used viruses to make the vaccine. And the kind of virus that they used to help make the vaccine was a very inefficient cancer virus. Only 1 in thousands of cells could turn into cancer based on the use of this virus. However, 10 to 30 million people were exposed to this vaccine. So if we see more cancer than we expect in those 10 to 30 million people, then we can blame the poliovirus vaccine for causing their cancer. This was a real concern and a real worry. But at the end, what they found was those 10 to 30 million people actually did not have increased cancers compared to the people who never got the vaccine. So viral vaccines are not very good at causing cancer, fortunately. Viruses are also used for gene therapy. So gene therapy is when, some people are born with very serious diseases that only one gene is either mutated or just not working right. So if you can give the correct version of that gene to that person, then maybe you could cure the disease. So you want something that can deliver the correct gene and integrate that correct gene into the DNA, into the host cell DNA. A virus sounds like the perfect vector to be delivering the good gene that you want. So they did this in France for a severe immunodeficiency disease called SCID. And they used a murine leukemia virus to deliver the good gene. Well, it did a pretty good job of delivering the good gene, but this murine leukemia virus actually integrated into unfavorable areas in the host cell DNA, and 4 of the children ended up getting leukemia. So this is because the viral gene, the viral vector, the thing carrying the good gene, inserted into a not good part of the host cell DNA. And so because of this, there has been a lot of research and development into picking viruses that don't do that as much, or picking viruses that do that in a much smarter, more controlled way. Talking about human tumor viruses, there are a couple different principles of human tumor viruses that are worth noting. Some of these human tumor viruses cause cancer directly. They carried the oncogenes. HPV, human papillomavirus, carries oncogenes, so it has genes that are cancer promoting. Sometimes it's more indirect how the human tumor viruses cause cancer. They don't carry oncogenes, but they cause a lot of inflammation, chronic infection, and just they're there for a long time and things aren't normal. So it makes it such that mutation can happen more easily, and then that's how you can get cancer. The other principle of human tumor viruses is they work pretty slowly, latency. And so the viruses, they don't want to kill the host cell quickly because they want to use the host cell and use the host cell machinery to make lots of copies. And it's a very nice home for the virus. And so they don't act quickly. And because the viruses are sort of hiding in the cells and not doing too much too actively, if you were to give an antiviral, it doesn't usually work on shrinking cancers that have already started. An antiviral, something that works against the virus, can prevent the virus from infecting new cells. But once the virus is already hiding in the cell, the antiviral therapies don't work very well at killing or shrinking those cells. And human tumor viruses are very good at hiding from the immune system. These next couple of slides are pretty complicated. I'm not going to go into detail. Just the main thing is this, the take home. Tumor viruses infect cells, and then in the cell, they cause a whole bunch of different responses. So the effects of the viral infection are things like it promotes mutation, it can promote cells to grow, it can make the DNA unstable, it can make transformation happen, chronic inflammation. Viruses cause a whole bunch of problems in cells. And a lot of the viruses actually activate very popular pathways that have already been implicated in cancer. So I won't go into the details of that either, but a lot of cancers activate p53 and retinoblastoma pathways, and viral tumor viruses actually activate cancer pathways as well. What we know about people who are immunosuppressed is that people with immune suppression actually are more likely to get cancer. So HIV patients have more cancer, and organ transplant patients have more cancer. Immune therapy is something that has a lot of potential to help virus-associated cancers. So a very popular type of immune therapy would be vaccination. If you give a vaccine, it can prevent a viral infection. There are viral vaccines to a couple human tumor viruses, including hepatitis B and human papillomavirus vaccines. Again, same thing. They prevent the virus from infecting new cells, but they don't do much about the cells that are already infected. They can, you can also give other types of antiviral treatments that could actually treat an infection before the infection can stick around too long to cause chronic infection, chronic inflammation, and then eventually cancer. So you can give a therapy that's an antiviral that would hopefully prevent the virus from sticking around and giving the virus the time it needs to transform cells. And then something a little bit more experimental that we're working on currently now these days is how can we actually use the immune system to try to shrink or kill the tumors that are already there? Vaccines won't do it, but can we use the immune system to try to go after the cancer cells that have virus in them? This is again a summary of the 8 tumor viruses, and then this time on the right I have the mechanism by which the virus is thought to cause the cancer. This is just here for your interest one day, not too relevant to my talk, just a lot of details. The second part of the talk is to talk a little bit about HPV, because it's the most prevalent tumor virus, and it's the cause of a rising cancer epidemic. About 5% of cancers worldwide are associated with HPV. And like any other virus, HPV has two things: a protein capsid and DNA. The DNA for HPV encodes early proteins and late proteins. And the cancer-causing properties, the oncogenes, all of the bad stuff cancer-wise that HPV does is related to the early proteins. The late proteins are just proteins that make the capsid, the protein coating shell. So the late proteins don't really have any role in causing cancer. The cancerogenic properties of HPV come from the early proteins. The life cycle of HPV. So HPV enters into a broken or denuded epithelium. So the skin surface or mucosal surface has to have some sort of trauma or opening. And then the HPV wants to go all the way down to the deepest part, the basement membrane, a place where it can hide. It will infect the cells. And that whole process involves the late protein, the capsid proteins, the L1 and L2 capsid proteins. So none of this getting into the basement cells is the cancer-causing part. This is just the infection part. And HPV is going to hide in the basement membrane cells for as long as it wants to. And if the infection stays long enough, eventually what it will do is it will cause replication and more virus to come out. But the viral particles come out at the surface. So because it's coming out at the surface, that's why HPV is very contagious through contact. But it's also why, because the viral particles are not entering into the bloodstream at the bottom, that's also why you can have an HPV infection for a really long time and the immune system never recognizes it. There are certain properties of cancer cells. We went over them in the first slide that cancer cells do this. They grow uncontrollably. They grow on top of each other. They don't listen to normal signals. They can travel. They can move. All of those properties come from the early proteins, the E proteins from HPV. Then there are the more subtle things about how, with all of these activation of all these pathways to make bad things happen for very clever reasons, the immune system never figures out what's happening, and HPV can basically evade the immune system. It can silence activation, it can hide, it can do all of those things. Most people will eventually clear an HPV infection. So it can linger and hang out, for a year or two, but most people will eventually clear it. Once that infection is cleared, basically the way it works is the immune system cells in the area of the infection recognize that there's an infection. It'll take that information to the lymph nodes, and then the lymph nodes will activate the effector cells of the immune system so that the immune system is now activated, saying we better go to this location and kill off these infected cells. And it can also create antibodies that will basically try to recognize free viral particles quicker to prevent an infection. But of note, these antibodies can only prevent infection. These antibodies are not going to kill the cells that have been infected. So if the HPV infection persists, meaning the immune system didn't take care of it, then eventually it can cause transformation. And so that the normal cells can start looking abnormal. very gradually, a little abnormal, low grade, high grade, a lot abnormal, and then full on cancer. It's traveling. But although cervical cancer is the main cancer associated with HPV, it's really just the tip of the iceberg. Actually now, HPV is causing more oropharyngeal cancers than it's causing cervical cancers. And part of the reason for that is because cervical cancer is, it has a screening test. It has a pap smear. And so pap smears are able to identify the HPV causing the low grade precancerous lesions. So with pap smears, you can take care of the issue before it ever got to the invasive cervical cancer. But there is no screening test for the throat for oropharyngeal cancers. Like other cancers, the pathways that are upregulated in HPV tumors are P53 retinoblastoma. We can check these with basically doing various types of lab studies on tumor specimens. I won't go into the detail of that, but this is something we do not just at MSK, but really pretty much every hospital in the country. We'll take a sample, a biopsy of a tumor, and then do some molecular work to basically look for HPV signals in these tumor specimens. So HPV positive oropharyngeal cancer, OPC, is actually increasing in young men. It used to be thought that head and neck cancer patients were older smokers who drink heavily, but actually now these days it's men in their 50s, and the amount of cancer you'll see in like the blue line is going up, increasing over time. HPV negative cancers, the ones that are caused by smoking, are actually decreasing because people are smoking less. The good news, though, with HPV positive oropharyngeal cancers is that their prognosis is actually a lot better than HPV negative cancer. So you will see the stars on the blue lines. Those are HPV-related oropharyngeal cancers that were treated with chemoradiation. And you could see that the chance of cure, if you look at the 0.9, it's around 90%. So these cancers, even though they're cancer, and even though it's caused by HPV, these patients are actually much, much, much more likely to be cured. So as a physician who treats these patients, I am very happy when I see that the patient has an HPV positive oropharyngeal cancer, because I know that they're most likely going to be okay. The way we treat HPV positive oropharyngeal cancers is tough. It's rough. It's 7 weeks of daily radiation with chemotherapy given at the same time. That's the most common way we do it. Sometimes it's surgery followed by 6 weeks of radiation. The prognosis is excellent. We're likely to cure them, but some of these patients for the rest of their lives, which could mean decades, for decades, food will never taste the same. They have dry mouth. They don't enjoy eating. Some people say it takes me, an hour to eat a hamburger, and it used to take me 10 minutes. And so they get depressed because they don't want to go to restaurants, or they don't want to eat around other people because it's difficult and embarrassing. So this treatment is really tough. It works very well, but you pay a price. And so some of the research going on at MSK within the group I work at is, well, how can we reduce the treatment, make it less severe, less side effects for HPV positive oropharyngeal cancers? We want to keep the cure at 90% or above, but we're trying to cut the treatment in half, maybe 3 weeks instead of 7 weeks, to decrease the side effects, to give them cure and good quality of life. Other things that we want to do is use personalized medicine. So if HPV caused the cancer, then maybe does HPV DNA levels, does that let us know how the cancer is doing? If the HPV DNA levels are low and they stay low, maybe that's someone who's cured. But if someone has HPV DNA levels that are rising, maybe that means something's brewing. That's another trial that we have at MSK. So we have a lot of really special things going on. In the interest of time, because I want to get to some other stuff, I'm not going to go over this, but there are a lot of things that we know and don't know. Even though we have tons of research and lots of smart people all over the world working on this, there is still so much room for young, creative people like yourselves to learn about this stuff and get involved. And, right now, due to COVID, we don't have as many, volunteers. But if anybody's interested in this stuff and wants to volunteer at some point in the future, when we're able to open up again, I'd be happy to set you up within our group. But here are a couple of the most common questions that are asked by my patients, just so that if you guys know anyone, you can say, well, we talked about this at the HOPP SSP lectures. Are partners of HPV positive cancers at increased risk of an HPV infection or cancer? With regards to the question of if your partner has an HPV-related cancer, are you at increased risk of HPV infection? We pretty much believe the answer to that is no, because most of the time what happened is the person had the HPV infection a long time ago. They are not actively shedding virus, and so it's unlikely that they're going to actually give their partner another HPV infection. However, with regards to the question of are partners of HPV positive cancer patients likely to get an HPV positive cancer, the short answer is we're not sure. There have been several studies that tried to answer this question. Bottom line, half of them said, yeah, we think there's a slightly increased risk of HPV-related cancer in the partner. And then the other half of the study said, nope, no increased risk. The absolute increased risk in terms of percentage is on the order of 1 to 3%. So not a big increased risk for the partner. And that's something that a lot of the, husbands or wives end up finding, reassuring. But it's a common question that comes up. People ask a lot about antibodies and vaccines. And so basically, I'm not going to go into all of this because this is a little too much, but the bottom line is they have found that people with HPV positive head and neck cancers do have a lot of HPV antibodies. What we don't know is if those antibodies just mean that their immune system is hyperactive, or if it actually means that these antibodies are going to protect them from a recurrence of the cancer. So we still have a lot of stuff we need to learn. The final part, this is the part I think is the most valuable for the high school students. So prevention is the best medicine. There is no cure for HPV infection because most of the times you have no symptoms. Over 90% of infections will actually clear on their own within a year or two due to the immune system. HPV can cause warts, but these oftentimes tend to come back even if you cut them out or freeze them. And do you know why? It's because the HPV likes to hide in the cells in the basement membrane, very deep inside. So even if you cut or freeze, you may have not gone deep enough to where the virus is hiding in those basement membrane cells. So really, prevention, there's no cure for the infection. So prevention is the best medicine. And that means vaccines. Remember I told you that all of HPV's bad behaviors come from the E, early proteins, and that the L, or late proteins, are basically capsid proteins. Well, all the vaccines that are currently in use now are only targeted against the late capsid proteins. That's probably why it doesn't work so well on treating an infection. That's probably why the vaccines really only work for prevention. Here are 3 vaccines that are available for use in the United States. Won't go into detail other than just saying this. There are about 200 different human papillomaviruses. The bivalent vaccine Cervarix targets 2, HPV 16 and 18, which actually is implicated in about 70% of cervical cancer. The quadrivalent Gardasil targets HPV numbers 6, 11, 16, and 18, and the nonavalent Gardasil 9 targets 6, 11, 16, 18, 31, 33, 44, 45, 52, and 58. So, all 3 of them are good. All 3 of them are about 90% effective at preventing cancer if you didn't have the infection at the time that you got the vaccine. And just to kind of show it to you in different ways. If you got the quadrivalent, so the nonavalent, the vaccine against 9 different viruses, there's not as much data because that's newer. But the quadrivalent against the 4 different viruses, 6, 11, 16, 18, if you had no HPV and you got all 3 shots, you're looking at about 90% effective at preventing cancer. If you got only 1 shot, or at least 1 shot, and you did not have HPV at the time of vaccination, it's not quite as good, but it's on the order of, let's say, around in the 70% range. So you really want to get all 3 shots to have the full efficacy, but even getting 1 shot is better than nothing. And if you had HPV, like you already had the infection, but you said, hey, I want the vaccine, it doesn't work great, maybe on the order of 30%, but it still does something. It doesn't do nothing. And so, I think it's great to get it even in that situation. There are a lot of guidelines, and basically in the United States, the CDC, this vaccine is only approved up to age 26. So if you're older than 26, your insurance is not going to pay for the vaccine for you. Hey, wait a minute. Even if you had HPV already, look, I mean, the vaccine does something. It's helping about 30%, but this is the way it is. If you're 26 or older, you got to pay if you want the vaccine. And part of the reason is in the trials, they didn't really study that many people older than 26, so they don't really know how well it works. And the assumption is if you're 26, you've already had the infection, so they don't know what the point is at that point. But there is, lately, more recently, there has been plenty of data saying the effectiveness is still something in people who are older. And if you're a 30-year-old, but you never had HPV, the vaccine is still 90% effective. So actually, my patients all the time, if they don't have HPV, they are paying out of pocket to get the vaccine because they want to do anything they can, to lower their risks. And I'm sure those of you who are watching the news have heard about herd immunity. If you give vaccine to some people, but not everyone, does that benefit the other people who didn't get the vaccine? And the answer with the HPV vaccine is yes. You only vaccinated the people of that younger age, but in the US and Australia, there was a 17 to 49% decrease in HPV infection, even amongst people who never got the vaccine. Unfortunately, globally, only about 6% of eligible females had gotten the vaccine by 2014. I'm going to ignore the thing. The thing in Sweden basically showed the same thing, which is young or old, the vaccine helps. It helps young people more than older people, but it really does help everybody. The cure rates for cervical cancer are excellent if it's early stage disease, 92. But once it gets a little more advanced, cervical cancer is very aggressive. So it's really important to do prevention. Because this was a talk on my specialty is throat cancer, it seems like the HPV vaccine which was intended to prevent cervical cancer, seems to actually decrease the amount of HPV in the throat as well. So we have a lot of hope in believing that these vaccines are not only going to hopefully one day get rid of cervical cancer, but get rid of or really reduce throat cancer as well. The US is a little better than the rest of the world about vaccine uptake, but we're not great either. So the left is how many people have gotten one dose at least. And then the right, figure number 2, is how many people got everything, all the doses they were supposed to. And you could see it's not even half amongst women and way less than that among men. So vaccines are preventative. The vaccines that we have now are all against late proteins. They prevent infection, but they don't treat infections or cancers that are already there. But there's a lot of research against trying to make vaccines against early proteins, and hopefully those vaccines against early proteins, maybe those will work against active infections or cancers. Bottom line, the most important thing, whether you're vaccinated or not, you still need cervical cancer screening. Even though the vaccines are over 90% effective at preventing cancer, if you got them on time when you were young, before you had HPV exposure, despite that, you still have to be screened the same way as if you were never vaccinated. I'm going to go into these 2 food for thoughts because I think they're relevant to what's going on in society today. And then I'll be done in 5 minutes, and then I'm happy to take questions. Here's food for thought number 1. In 2019, a highly infectious virus was discovered. It took over the world. And we learned that its components included a single-stranded RNA genome, a spike glycoprotein, a membrane glycoprotein, an envelope and nucleocapsid proteins. Sounds familiar. If hypothetically, many years later, we were to discover that latent infection with this virus caused cancer, what are the mechanisms by which this could have happened? Well, this virus could have carried an oncogene, its own unique oncogene. Or the virus could have integrated into the DNA into an unfavorable location and activated an oncogene that was already there in the host cell, or just caused a mutation and something new and bad to happen. Or latent viral infection, the virus hiding in the cells and not doing much for a long time, could cause chronic inflammation and genetic instability, which eventually can lead to cancer. Food for thought number 2. In response to the medical, social, and economic burdens due to the new virus, 2 vaccines were developed. One vaccine carried DNA or RNA against the spike protein, which is against the coating, the capsid. And it was carried within a virus vector. So a virus has DNA against the spike protein, and you give this virus containing that spike protein DNA, that's your vaccine. The other vaccine is a lipid particle that contains RNA for the spike protein, and you give this lipid particle. Which one would be more likely to cause cancer? Well, the lipid particle containing mRNA cannot cause cancer. So that one definitely not. But even the virus vector, if you mutate the virus so that it cannot integrate into the host cell DNA, then that one will not be able to cause cancer either, as long as you take away the ability for the virus to integrate into the DNA. So the flu shot, the measles, Ebola vaccines, the J&J vaccine, all of them use a mutated viral vector that cannot integrate into the host cell DNA. And that's why we know it's safe. But Moderna and Pfizer use the lipid particle. Okay, I am done with my talk. I guess I will open it up to questions.
Great. Thank you so much, Dr. Kang. We do have a couple of questions. One was in regards to internships and projects. So a high school student, they wanted to know about potential research ideas they could carry out with their school lab facilities to do a research paper. Would this be a question that's best to contact you directly with any questions, or did you have any ideas in mind that you'd like to share with them?
Yeah, I mean, so the thing is unfortunately right now due to COVID, we haven't, I don't think anyone at the institution has been allowed to accept research volunteers outside of MSK. But, you know, usually we just have people email us. So maybe later you can give them my email, and anyone interested, I'll basically just make a list. And as soon as I'm told by admin that we're allowed to accept volunteers, we basically have I do head and neck cancer research, and we have a group of about 8 of us who, you know, have various different projects, and we mentor people all the time. And, you know, it's a lot of work to do research. We don't do as much of the lab research. We do more clinical research. So as an example, I told you guys that at MSK, we're trying to reduce the side effects and reduce the toxicity for HPV positive oropharyngeal cancer patients by cutting the radiation in half from 7 weeks to 3 weeks. Well, I am working on a similar project for HPV negative cancer. So there's all sorts of projects. We're working on a vaccine trial as well. So there's so many things that we're doing all the time, and we love volunteers. It's just the past year and a half, we've been limited by the fact that due to COVID, we're not allowed to have people come in to meet with us, and they don't get access to like the computers and all those things that they need in order to do research.
Okay, great. The next question they're asking is, why is vaccination after infection less effective?
That's a great question. So if we're talking about HPV, the reason is because basically the HPV virus likes to live and hide in the cells in the basement membrane. And while the virus is hiding in the cells in the basement membrane, a lot of the times it's doing nothing. It's not actually trying to replicate the virus. It's not making more virus particles. It's actually just hitchhiked in that cell, and it's doing absolutely nothing. And so when it's hiding in the cell, then nothing's coming outside. So if you think about an antibody like the letter Y, think about the triangle part of the Y, the upper part of the Y. That needs to be able to see HPV virus L1, L2 capsid proteins to attack and then say, hey, clear me up. But if those viruses are not outside the cell, if they're hiding inside of the cell, the antibodies won't even see that there's anything around. And so you can give these antibodies or you can give the L1, L2 protein and generate antibodies. But if you don't have virus outside of the cell, and if it's hiding in the cell, then the antibody won't see it.
Okay, great. Thank you so much, Dr. Kang, for your talk. Thank you to all of our registrants for attending today's seminar. It's been a pleasure hosting you all every week, and we hope you continue to be in touch for upcoming events. Thank you again. Have a good day, everyone.
Thank you.